Welcome to the Neural Implant Podcast, where we talk with the people behind the current events and breakthroughs in brain implants in an understandable way, helping bring together various fields involved in neuroprosthetics. Here is your host, Laden Yurachek.
Hey everyone, and welcome to the Neural Implant Podcast. Today's guest is a special guest. It's Ed Boyden of MIT. Now he's really cool because he co-invented optogenetic tools. So these are tools, these are basically proteins which allow the activation and silencing of neural activity with light. So you don't have to use electricity, you don't have to use anything else, but you can use light to do this. Now this has actually been huge in the last decade plus, and it's actually made him very, very well respected, and it's won him a prize. It's won him the Breakthrough Prize in Life Sciences. Now, with this prize came $3 million as well. And if you listen to the episode, he promises me, for the Neural Implant Podcast, he promises $1 million for a new microphone. So, take a listen for this part. Anyways, this was really interesting. He also has another project, and this is on expansion microscopy. So this is basically expanding the neurons and expanding all the proteins and everything inside to such a level that it can be imaged much, much easier. So it expands it by 100 times, and then you can see what's going on, a little snapshot in time. So this is really cool. This has been a very interesting talk. Well, there was rumors online that he could be, next in line for the Nobel Prize. Why not? If he's won a $3 million prize, why not win a $1 million prize? So hopefully you guys will enjoy this, and we'll talk a little bit more on the other side.
I was very philosophical as a kid. I really wanted to understand the meaning of life in a very concrete way. And so I started college pretty young. I was only 14 years old, and I worked in a chemistry lab that was trying to create life from scratch, which of course didn't work, or you'd have heard about it by now. I gravitated to a lot of these questions that are sort of the border of science and philosophy, if you will. And I moved on to study physics. And finally decided that the brain was where the action was. And so coincidentally, all of my training in chemistry and physics perfectly prepared me to go develop technology for the brain. So I started grad school at Stanford in the year 1999. And around that time, I met my fellow student, Carl Deisroth. He had just finished the PhD part and was doing the MD part of his training. And we started brainstorming. And at the time, Carl worked on transcranial magnetic stimulation, or TMS. A way to activate the brain, but not with cell type or single cell precision. And we started brainstorming, just going through all the laws of physics, thinking, how could you stimulate a cell or more precisely, ideally even a single cell, or at least one kind of cell? And after looking at mechanical and magnetic and all sorts of other ways of perturbing neural activity, we really honed in on light. And I stumbled across this paper which had come out from a group in Japan, and they had reported that one of these molecules known as opsins, 7 transmembrane proteins that respond to light by pumping ions across membranes. A lot of these come from archaea, single-celled microbes that live in very high salt environments. But one of these molecules seemed to work at low salt concentrations, like you would find in the brain. And so I wrote to one of the key discoverers of those molecules, Janos Lanyi, and he forwarded my email to a colleague, and that's how we started collecting these molecules from colleagues to try out.
And then you were able to put it into the brain. The neurons were able to accept it?
Yeah, so Carl started his residency training, and I went off to Woods Hole for a summer course. We actually didn't do much with the molecules right away. But in 2003, a paper came out describing a different kind of molecule from algae. And right away, we pounced on it and started messing around in the lab with this molecule, chenorodopsin-2. And basically, it worked on the first try. I put the molecule into, Carl put the molecule into some neurons, and I shined light on them and recorded them. And I still remember the moment. It was early in the morning one day, about 1 o'clock in the morning. And the neuron responded to light on the first try. And that was really a very visceral experience to watch, an engine of thinking and emotion of the brain respond to a force of physics. So
Yeah, that's pretty crazy. That's the beginning of a revolution, I guess.
Well, one that's just beginning. Optogenetics has been used in a lot of groups, but do we have a theory of the brain yet? Do we understand how the brain computes sensations or thoughts or feelings or actions? The answer is still no. And in my group, we're working on a lot of different technologies because, it's not a very good analogy, but maybe let's run with it a little bit. Imagine you're trying to understand and repair a computer. You need the keyboard so you can type information in, but you also need a monitor so you can watch what's going on. And ideally, you also know the wiring of the computer so you can have a concept of how information is flowing and being transformed. And so, my group is working now on all three of those kinds of technologies: control, but also imaging of real-time dynamics, recording of real-time dynamics, and then also mapping the wiring of the brain. And my hope is that in the coming decade, we'll start to see theories of how neural circuits work. And your podcast is called the Neural Implant Podcast. I would argue that neural implants are important, but you also got to know how to use them. Sometimes I tell people the hard part of neuroengineering is the neural part. Where do you put the implant? How do you analyze the data? How do you contextualize the neurophysiology as what's going on in a circuit with defined architecture and rules? And my hope is that through mapping, recording, and perturbing brain circuits, we'll learn the principles of brain operation, and that'll, empower us to build brain-machine interfaces that really can speak the natural language of the brain.
So are you saying that optogenetics is not fulfilling its potential yet? And what do you think is needed to be able to fulfill this potential?
Well, I think that it's very clear that controlling is only part of the story. You have to be able to map the brain and record the activity of the brain as well. And then ideally build a computational framework to help deal with all that data and integrate it towards theories of brain operation. So in our group, we're working on high-density electrode arrays that can record from perhaps thousands of sites throughout the brain, maybe someday millions of channels. We're also working on optical imaging methods that could let us watch brain activity in many neurons throughout a three-dimensional brain circuit and to record the neural activity in real time. We're also working on ways to map the architecture of the brain. That's a really difficult problem because the brain is a large object, but it's wired with nanoscale precision, and the connections are also nanoscale. I mean, this is sort of an estimate, but I think if you somehow could blow a brain up, a human brain, till it was the size of a city block, the connections between neurons would only be the size of grains of sand. So that tells you a little bit of how the differences in scale of comprehension have to be in order to understand how the brain wiring is generated and how it's used. So in my group, we decided, let's take this idea literally. People have been trying to zoom in for hundreds of years, and that's been difficult. Why don't we blow up the brain? And so we can now take a piece of brain tissue and we synthesize a polymer that's a lot like the stuff in baby diapers, except with a very well-defined and dense topology. We add water, and the baby diaper polymer will swell, but it's in a very even isotropic fashion. And so we can physically magnify brain circuits, and what we're finding is that we can actually detect wires and connections and with great fidelity. And our hope is sometime soon we could actually map brain circuits this way.
Exactly. So instead of a grain of sand, it would be maybe the size of, well, what you find inside of a baby diaper. And I was actually going to laugh at you, like that you took the inside of your kid's baby diaper, what you found in there, and put that in the brain.
Yeah, well, that's actually not how it happened. I always get asked the question, was it when you were changing diapers that the idea came to you? In reality, I was brainstorming with a postdoc in my group back in 2006, 2007. And just like optogenetics, we decided, let's just go through all the ideas of how you would physically magnify the brain and separate the molecules. At the time, I really wanted a solution to the molecular crowding problem, this idea that molecules are so packed together that if you wanted to really image them, you need to make room around them so that you can bring in tags that indicate which molecule is which. Molecules in the brain, all look kind of similar from a physics standpoint. You have to use chemistry to tell them apart. And so we decided to try to decrowd the molecules from each other. But this is right around the time that super resolution microscopy came out, and we decided, it's a pretty crowded space, no pun intended. They'll figure it out. It wasn't until 5 years later when we tried to use some of these tools and we realized, what? If you're trying to map a large-scale three-dimensional brain circuit, we're gonna need a new technology. They were not fast enough and to map these large-scale 3D circuits that are so important in the brain.
And it's like a snapshot, right? Like you couldn't see movement or anything like this because obviously when it expands, it takes it out of its usual place, right?
Correct.
Cool. Well, this is really interesting. I mean, I like this, finding a present inside your kid's diaper. But I really like the optogenetics more, and especially with the neural implants, because you can introduce and read out information without really causing too much noise, without causing, I don't know, stray electric fields to light up other neural pathways. So that's really the benefit, I think, of optogenetics, is that it can be extremely precise and without any noise as well, right?
Well, there's always noise. I mean, any physical method will have, because we're not at absolute zero, some degree of noise. But what it does allow you to do is to activate individual cells or sets of cells of a given kind. And that's important because the brain is made out of thousands of different kinds of cells. There are excitatory and inhibitory cells. There are modulatory cells. There are cells that are small and star-shaped, and there are cells that are large and project around the entire brain. And so, being able to tell these cells apart and then activate them and figure out how they each play roles in this, symphony orchestra brain computation, if you will, is really critical. And we've now sent these technologies from our group out to literally thousands of groups around the world, and they're being used in papers coming out every day to activate and silence different cell types.
And so would you say that's one of the biggest challenges, is identifying the different types of cells with this research?
Well, that's another reason why expansion is so helpful. So when we expand cells, we decrowd all the molecules from each other, and now we can run interesting chemical reactions inside the cells. So, for example, one thing we're working on, collaborating with George Church's group at Harvard, is to get sequencing, nucleic acid sequencing, working inside of expanded brains. So imagine you could expand a cell, separate all the gene expression products, the RNAs, as they're technically known, and then you could sequence them right there. And that would allow you then to understand the recipe of a cell, what genes are on and which genes are off. So now you could get a list of the cell types in an actual circuit where you can also look at the map of the circuitry.
Yeah, that's a pretty futuristic stuff, I guess.
Well, we're doing it in the lab now. It's an actual project, and we have students and postdocs working in both my group and in George Church's group on this project. It's actually funny you said of the future, because I sometimes get emails from science fiction authors saying, "Oh, what do you think of this idea?" And I'm like, "We're actually doing that already. You have to think harder."
Actually, we published that last year, so yeah, sorry. Well, actually, if you had a magic wand, what would you change in this field?
Well, I think that the way that biology and medicine research works, it's very hypothesis driven. It's governed by peer review. I sometimes think that what we need to do is to make the world safe for serendipity. If you look at the really big innovations, many of them were highly unpredictable, or they were hiding in plain sight, and it took somebody without the standard credentials, an outsider, to make it reality and to make it happen. So, I think that one of the things I'm often looking for is how do we take the ideas that fall through the cracks and make sure they happen? I was an outsider when I came into neuroscience. This idea for expansion microscopy, where we blow the brain up to make it bigger, people didn't believe it for the first several years. They're like, yeah, right. You can evenly blow a brain up and make it 100 times bigger in volume and still preserve all the arrangement of molecules. It wasn't really till our fifth paper was coming out that people started to say, all right, maybe there's something going on here. Meanwhile, of course, hundreds of groups had sent people either to our group or to one of our classes that we taught to learn how to do it. So I often think about how do you make the world safe for serendipity? And this is not a unique complaint. The guy who won the Nobel Prize for crystallizing G protein coupled receptors lost his funding and had to work as a hospital employee to pay the bills. Or the guy who cloned the gene for green fluorescent protein ran out of cash and ended up eventually leaving science and, if I recall, driving a rental car shuttle. I mean, you see these kinds of things all the time where the really big serendipitous insights they sound so foreign to people, and sometimes they fall through the cracks.
So I guess legitimizing citizen science and maybe giving them a platform so they could publish, quote unquote, right?
Not really. I think publishing actually is already being addressed in many useful ways, preprint servers and all sorts of stuff like that. I think what's often tricky is somebody has a really good idea and it's hard to get resources to make it happen. That hasn't changed in a long time. Publishing, you could argue, is undergoing a renaissance thanks to preprint servers and open access and all sorts of stuff. But funding is evolving a bit slower.
That's kind of funny that that you mentioned this because, I've been kind of sitting on an idea, basically hydrogel coated brain implants, and you can use essentially, 2-photon polymerization. So like get really, really fine structures on there. And I've been sitting on this idea for like 2 years and I just haven't been able to, get it out. And I don't know. I think of course, the theory has some holes, or the idea has some holes. It can be definitely improved. But I would second this. It's sometimes difficult, for some, unless you are a professor or unless you have a position, you might not be able to add anything. Also, the problem with this idea is it's very expensive.
Well, these people I'm talking about were professors. They had published. They had government grants. It's
so nobody's safe.
And yeah, even they could. I already had gotten tenure by the time that we were, gearing up to write a lot of grants on this expansion microscopy idea, and still it didn't matter. So yeah, I don't think it's a matter of credibility. In fact, maybe that's the problem, is that it's so hard to judge a biomedical idea, and so a lot of people try to judge them based upon opinions rather than upon logic and first principles thinking. It's very hard to do, and maybe it requires different sets of evaluators who work across disciplines. I'm one of the 4 interviewers for the Hertz Foundation, which gives out 5-year graduate fellowships. And so people have to get past me or one of the other 3, and we interview in pairs because we want to make sure that we're always taking different points of expertise. And one of the things I love about Hertz Foundation interviewing is, suppose somebody gets a couple different scores, we rank people by their best score, because you can always find something wrong with somebody's knowledge or a gap in their expertise or a flaw in an idea. But we want to know what makes somebody special. Are they really going to revolutionize the field because they have that extra spark of creativity or rebelliousness or out of the box familiarity with different fields?
So you're saying if they just wow you with a magic trick, then they're in, right?
No. We have a lot of different criteria. No, we want to know. We want to know about depth too. Superficiality is what did Einstein say? Beware the seduction of superficiality. We did a magic trick. You better be able to explain it at a very deep level, and we'll keep asking questions about it in different ways.
Well, I guess this kind of dovetails into my next question, is that many listeners to this show, and they've written me and they said, they're just starting out in this field, and they're looking for recommendations for neural implants, neuroscience. What would you recommend besides going for this Hertz Fellowship?
Yeah, that's a long topic. The way our group works is we practice a very unique way of problem solving. Most groups have an area of expertise. They're the best at carbon nanotubes, or the best at nanodiamonds, or they're the best at certain circuits involved with Parkinson's disease. Our group does not have an area of expertise per se. I train very broadly: chemistry, physics, neuroscience. I prefer to think we have a strategy focus. And here's our strategy. We start by picking a very deep problem. We don't take requests because we don't want to address the problem at the level that we understand it now. We often want to dig one level deeper, right? What's the old saying? If Henry Ford did what people asked him to do, he would have tried to breed a faster horse, right? Because his customers didn't have the concept of the automobile in their head, right? So the first is we try to understand a problem at a very deep level. Second, we think backwards from that problem, and we try to survey every possible strategy to go for it. Like I told you earlier, optogenetics started by going through all the laws of physics, and expansion microscopy started by thinking about every way you would swell a biological specimen. So we try to have every possible idea, and we have strategies for going about that. And then the third stage is what I call wisdom gathering. We try lots of stuff out, and most of it fails, but each failure teaches us something new that most likely nobody's seen before. And then from that wisdom, we go to the design phase, where now we know what we're doing, and we make the actual invention. And if all that works, then something that I sometimes call the principle of applied laziness kicks in, which is that the actual work, once we reach the design phase, can go pretty quickly, because now we kind of know what we're doing.
I'm very curious about this wisdom gathering. What does that look like?
Oh, well, people try things out, and a lot of it fails, and they notice things that people haven't noticed before. But over time, we can connect the dots across these, and that leads to a powerful new design or a new invention.
Yeah, my master's in Germany had really an amazing thing. It was called Beer Hour, and every Friday at 5 p.m. there was free beer, and everybody from all the labs would meet up and discuss basically their problems. Like, this is not going, and it's amazing. Like, there was some papers published in the journal Science that came from these discussions and this idea sex. So I think this wisdom gathering maybe. In my opinion, maybe it's the most important part of this whole process, because, as you said, the design process may be actually very intuitive.
Yeah, and by wisdom gathering, I don't mean things that you could read about or talk about. I think those are, if that's all you end up with, you're never going to know more than other people. But if you can really try something and notice something with your own hands and your own eyes and experiment, that's often the key. So, some kind of, playing around in the lab and you see something that nobody's seen before, and now you can connect dots in a different way.
Yeah, so I'm kind of wondering. I mean, it seems kind of like a chicken and the egg problem. Like if you're working at a level beyond what everybody else is doing and like solving, I guess, the big problems, then that wisdom gathering sounds especially difficult, actually.
Well, that's again why I think wisdom gathering has to be hands-on. You have to be trying things out and noticing things that nobody's seen before.
So what do you think the future is in neural implants? Like, what do you see in like 5 or 10 years?
Well, lots of people, even entire companies now, are being formed to look at making electrical implants. There are lots of people interested in optical imaging of the brain. Basically, you need a way to get information into the brain and a way to get information out of the brain. And there's a lot of effort going on in both areas. I mean, again, I think you can kind of go through all the laws of physics. There's electricity, there's light, there's magnetism, there's ultrasound, there's radio. And then the question then is, how does each of those things couple to brain activity? And then what are the constraints? Some might be more invasive, some might be less invasive, some might be more precise, some might be less precise. But what I see is the field pushing on all of these directions, and they're going to find different uses. And I think that's very exciting.
So it'd be like light input, radio output, ultrasound, I don't know, modulation or something like this, right? Like you'd use all these different things to achieve different goals, right?
Well, different people might pursue different things, right? I mean, again, to go back to an earlier topic, I like to think backwards, right? What's the problem you're trying to solve? Then design your approach, right? And I think that that kind of problem-solving strategy is where the real power comes from.
I guess that's the takeaway of this interview is work backwards and do the unconventional thing to get unconventional results, maybe, right?
Well, I don't believe in unconventionality for unconventionality's sake. I think you should do what solves the problem.
Yeah, that's true. Well, okay, so I want to talk also about this breakthrough prize that you won last year. How was that? You won $3 million with that too, huh?
That's right. Yeah. So the jury, which is all the previous breakthrough prize winners over the last several years, picked Carl and me to win breakthrough prizes. They actually gave us each $3 million. They decided that we had each contributed a major component to it, and therefore they didn't even ask us to split the prize. They actually gave us each one prize. So that was a big surprise. Usually people wait until you're much older to give you these kinds of things. And it was very exciting, and I'm very happy that it raised the profile of neural tools. A lot of the previous prizes were given for more basic biology.
Here's to everybody who says that science is not a lucrative field. And actually, you're in luck because Neural Implant Podcast just opened up donations today. I think 1 million would be enough. 1 million would be enough for a new microphone. Okay. Well, and then the last question too. I heard some, chattering on the internet about a Nobel Prize. What do you think about this?
Well, I think that Nobel Prizes usually wait till many years after innovation to make sure it's the real deal. What if optogenetics is replaced by something tomorrow and it's obsolete? That probably won't happen, but it's but I also think that one of the things that's emerging is also the arc of these technologies involves so many people as well, right? And if you look at other areas like super resolution microscopy or the green fluorescent protein or other technologies that have been recognized by the Nobel, these are large collaborative endeavors, involve many people. So the Nobel, as only has a limit of 3 people. And so one of the big questions, I think, is there a way to recognize the nature of collaboration? And the original discovery of the molecules, the class of molecules that Carl and I put into neurons was in 1971 when it was reported. And then over the years, people found molecules that pump chloride and molecules that, move sodium and other things like that. And so if you think about the arc of history in neuroscience and neural tools, these stories are involving all sorts of interesting traits, right? Curiosity about the natural world, the desire to explore the brain, the desire to invent, the desire to apply. And so that's a question that I ask a lot of people nowadays is there a way to recognize the entire arc of an innovation? And prizes, I think, can contribute to that, but they can also, I think, potentially emphasize one part of the story more than others. And so one thing I think a lot about is, what's the nature of scientific storytelling? How do we help people understand how science is actually done? Taking a step back from the topic and thinking about the bigger picture for a second, if you look at how science is taught in school, okay, so and so they did DNA, and so did genetic code, but the actual odysseys of discovery and the drama and the excitement and the struggle and the frustration and all that usually doesn't get captured. I think of science as an adventure story, and it's about as dramatic as anything gets. There's gambling, there's betting on an idea, there's failure, there's and so that's something I've been thinking a lot about, is how do we tell the story of science in a way that really captures the excitement in a way that maybe hasn't been captured before.
If I see further, it's because I've stood on the shoulders of giants. But actually, it's not giants. It's more like a giant heap of hobbits. And so, which three hobbits do you choose, right? So, yeah, I mean, it's a lot less sexy story, I guess. But as you said, actually, it could be more interesting because there's gambling and betrayal and all this kind of stuff and everything that makes for a good story. So, yeah, very cool. Do you have any recommendations on who we should talk to or what we should read?
Well, I would try for the podcast, I think there's so many new people entering the field. It's always great to talk to people who maybe are, just getting started or up and coming or have a crazy new idea and maybe benefit from having exposure to a broader audience. There's lots of entrepreneurs entering this field too, which is really interesting. There are people who are really, trying to, and I think this is very exciting, turn neurotechnology into a more mature industry, which would be very exciting. Is this like Silicon Valley in the 1960s, 1970s, or is it too early? And my bet is that we're on the cusp of a discipline that's going to mature in very interesting and somewhat unpredictable ways.
Yeah, so the movers and shakers, talk to them, right?
Well, but the movers and shakers, you might not even know who they are necessarily yet, because they might have the big idea in their garage and you might not hear about it for 3 years.
The aim of this podcast is to hear about it first. Dr. Ed Boyden, this has been a pleasure. This has been really cool. Thank you for your contribution and for your continuing contribution. You're not sitting on your laurels, but you're continuing to, push the boundaries. So thank you so much for this.
Thanks. Well, I'm 10 years in now to my faculty job at MIT. For me, I think of solving the brain as a roughly 30 to 40-year plan. We have the tool era. We're now in the heat of that era. Then I think there'll be a second era, which will be the computational era, making sense of all the data. And there'll probably be a third era, which is, augmentation and implants and boosting based upon the principles that emerge. And what I hope for is a fourth era, which I sometimes call the Enlightenment Era. If we understand the nature of human strife and irrationality and violence and war and all that, can we take control of our destiny as a species, if you will? And I was at a dinner and a head of state was there. And he was lamenting that economics and psychology and all these fields, had not brought peace to the world. And he looked at me and says, you know what, it's got to be the brain. And at that point, he didn't know I was. He didn't know I was a neuroscientist. And I said, well, funny you mention that. That's exactly what, we want to do. And so I've been thinking a lot about that lately. Where does this eventually connect with philosophy and the human condition? And as I mentioned earlier, that's kind of how I got started in this as a kid, was, wondering, is there something that we can do as scientists that will contribute to human happiness and to human meaning?
I mean, my feeling is that the unlocking the secrets kind of takes away the mystery. And I mean, when we know what atoms really are, when we know why the sun goes up and everything like this, it's more, I guess, logical or something like this, and it's not like the Greek style, very interesting discussions and everything like this. So yeah, it's very interesting. I think it will be good to finally put some of these philosophical questions to rest. But yeah, maybe the wonder might go away. What do you think? Like, do you agree with me on this or no?
Tell that to the millions of people who went out for the eclipse on Monday here in the United States. I mean, I think that I have the opposite point of view. I think understanding something and knowing how it works increases wonder, because now you don't have to necessarily confront something with this sense of confusion and fear. It's something now where, wow, look at the elegance of how all that works, like clockwork. And I feel like the elegance of understanding, there's a beauty to it that's like music or like poetry. If you look at the heart of science, the most interesting scientific discoveries have increased our wonder, whether it's understanding, the nature of how life works. Who would have thought the genetic code would be spelled out in letters, coiled in a helix, tucked in every cell? And every one of your cells, if you were to stretch out that helix, it would be a meter in length, an enormous amount of information coiled up into this tiny, tiny cell. And to think that that information is handed down generation after generation through the millennia and through, even the millions of years, copying and changing and altering, I find that fascinating. And I I would be hard pressed to see how just saying, well, here's a mystery, we don't understand anything about it, is that less wondrous or more wondrous than understanding this double helix of genetic code that's been passed down over the millennia? I think that it's incredible that encodes for an entire person, don't you?
Yeah, that's true. I mean, I guess the way you've said it also is a very interesting, a very engrossing story that is like, oh, I want to listen, I want to learn, hear more, and I guess that's what science should be. So instead of like, oh, here's this theory, I guess what you're saying is the truth is probably more strange, and once you know the truth as well, then you can play with it and actually like design and have fun with it too, right?
Yeah. Again, I think that again, the way science is taught in a lot of textbooks, you have these facts and they're to be memorized and whatnot. But if you understand the beauty of the truth within the facts, if you can understand the elegance of the understanding, I think that's where the true incredibleness of science comes out.
Science Poetry Slam. Okay,
there you go.
Got it. That's verbal confirmation. You and I are going to work on a science poetry slam. Okay, Ed, this has been excellent. Thank you so much for your time. I want to be respectful of your time. If people want to find out more about you, how do they do that?
Just Google me and you'll find all of my actual contact information. That's my actual email address and so forth.
Excellent. Thank you.
All right. Have fun.
Hey guys, hopefully you enjoyed it. I especially like the part where I guess he laments the, crystallized knowledge or crystallized ways of thinking of the scientists, I guess, in power. And it is kind of unfortunate, but I guess it is a reality that many very revolutionary and breakthrough ideas are shunned by the scientific community. And this happens all the time in all fields. And it's quite sad actually that this happens. So I guess this was the takeaway for me. This was really the takeaway for me for this show, was that, we should be a little bit more open-minded, and even if something sounds like it shouldn't work, it could still work. So another thing that I learned in this episode was that you should do the opposite. You should do the opposite of what everybody else is doing. Maybe start with the results and work from there. And this really seemed to be at the root of many of the great discoveries that Dr. Boyden did. So hopefully you guys enjoyed this. Hopefully you learned something new with this. I always love to have famous guests and not so famous guests too. I love to have both because I think it kind of brings it all together. And we can have citizen scientists on the show as well. So until next time, this is the Neural Implant Podcast.
Hope you enjoyed the show and were able to learn something new, bringing together different fields in novel ways. Until next time on the Neural Implant Podcast.